Today we'll be doing modern single-cell RNA-seq analysis using the Scanpy package or Scanpy package and also the latest Python tools. We are going to analyze 55,000 CD8 plus T-cells using the most modern Python tools available in 2025. We'll discover different T-cell populations, create beautiful visualizations, and do it all in just 30 minutes. And the best part, we are using the tools that are 10 times faster than the traditional methods. So we head over to the 10x genomics dataset and type in CD8 plus T cells. So it will show you 6 datasets, 1, 2, 3, 4, 5, 6. So I'm choosing this one because it contains 55,000 cells, which is fairly easy to compute on your laptop. And I have already downloaded this dataset. You can choose any sort of dataset for your calculations. I have already prepared analysis.py, a Python script to perform single cell analysis using the Scanpy package. And these are the requirements that you require, such as these are some of the latest packages like Marimo, the latest Scanpy, polars instead of the pandas, NumPy, matplotlib, seaborn, even the pandas also, because you need to convert into polars for the fast processing, and some other packages like UMAP, learn, and pyarrow. Okay, these are some of the requirements that you require. And so we'll be using these modern tools, right? So first, let's set up a modern Python environment instead of the old, Conda approach or pip approach. What we are going to do is we head over to the desktop directory where my data sets are. Then you require this particular package called UV. You head over to the GitHub, you type in UV, and you will see this package which is 10 to 100x faster than pip, as they have mentioned, and I have tested and tried it. It really is faster. You can install on Mac OS like this, or you can install on Windows by using this particular command. Okay, so I have already installed it, so let me show you how it works. So UV, if you type, then you have all these command lines that you can run for the package management or the Python environments. So the first command that you require is UV venv. So it will create a virtual environment for you, and it will tell you that you need to activate this with source venv bin activate. Okay, so you activate that. So when you head over to this particular profile, you will see that the virtual environment has been created. Okay, now if you need to install any package, then you type in uv pip install any sort of package. For instance, I need matplotlib. So if you type in, and you will see how fast it is. It is faster than pip package management. Okay, and also what you can do is uv pip install -r. I have this requirements.file, so requirements.txt file. You can do that, and it will quickly install all your packages, whatever the packages that I have written in the requirements.txt file. So it will install everything on that particular virtual environment. Now everything is set. So we'll be using not the Jupyter notebook, but instead of Jupyter, we'll be using the Marimo notebooks. These are the reactive notebooks. So when I change one cell, everything updates automatically. And we are using the latest Scanpy packages. Okay, so to open that, what you have to type is Marimo. If you type in Marimo, it will tell you all different bunch of commands that you can install or you can run. So Marimo, if you type edit and your any Python files .py extension, so it will open up on your browser, and it will show this particular notebook. Okay, so once you open this, so it will automatically try to run each and every packages. So that's the beauty of Marimo, is that it starts running when your analysis file or the .py file is ready to run. So it's much better. Or else you can tap in here. The cell is already, it says that cell is already running, so all my packages are getting installed. So we are using Marimo, so you can load by inputting this particular syntax, import Marimo as mo. That's a reactive notebook, and then your latest Scanpy package. And instead of pandas, we are using polars that will process the large data sets faster, and some of the plotting tools like matplotlib and cbon, etc. So once we load all the libraries, we are going to load our data set, which contains 55,000 CD8 plus T cells. We can use this particular syntax to load our data set. Okay, and we are making the gene names unique so that it doesn't repeat throughout the data sets. Okay, and you can see that the data set shape is around 55,000 cells and 33,000 genes. Whether the gene names are unique or not, yes, they are, because we're using this particular syntax to make it unique. And these are the first 10 genes that are displayed over here. You can print those information over here. Okay, so since we are loading 55,000 cells, which is around 116 MB data sets, it might take forever to load this data set in a normal computer if you are using pandas or any other libraries. But with the modern stack Python tools, you can load it much more faster, much more quicker. Okay, so that's the beauty of using the modern tools. Next, we move on to the quality control metrics. So this is a very crucial in single cell analysis, and we need to identify and remove low quality cells and genes that could bias our results. For instance, the mitochondrial genes, the ribosomal genes, hemoglobin, etc. You can input all sorts of low quality cells, and then you can filter them out or to detect them during the quality control metrics. The mitochondrial content that you put over here can be implied by using the notation as MT or MT minus CO1 and MT minus ND1. Ribosomal genes as RPS and RPL, and hemoglobin with HB and P. Okay, these are some of the notations that you can filter them out from your single cell data. The high mitochondrial genes, why they are important? Because this indicates the dying or stress cells, and typically healthy cells typically have less than 20% of mitochondrial gene expression. Ribosomal genes, these are the protein synthesis machinery. Very active cells have high ribosomal gene expression. So in the context of CD8 plus T context, these are activated T cells, which are the protein factories. Okay, now we are filtering them out using these notations, and you can see that we found 13 mitochondrial genes and 104 ribosomal genes, not a single hemoglobin genes from this analysis. Okay, and these are some of the QC stats where you see the counts. The total counts is 55,000, and number of genes by count is 55,206. And these are the percentile counts for your mitochondrial genes and ribosomal genes. Some quality metrics. Okay, now let's move on to the QC plots. You want to visualize all the data sets that you found from here, like the mitochondrial and ribosomal genes, etc. So you can plot them using this particular syntax code. So I'm using matplotlib for this, so it can print out all the rows and columns of that particular cell, right? So the the first plot is the UMI counts, and what you see over here in the x-axis is the total UMI unique molecular identifier counts per cell, and number of cells with that count. The Y represents the number of cells. The shape should look like a bell curve, like as you have seen, like this, as a bell curve. Around 4,000 UMIs per cell healthy for T cells. The median is 4,000 cells, which is absolutely fine for our data set. The second plot that you are seeing here is genes per cell. So on the x-axis, you have number of detected genes per cell, y-axis number of cells. So the range is 500 to 6,000 genes typical for the T cells, and median is 1400 genes per cell, which is absolutely fine. And you have seen that in the data set itself. In the description of the dataset, they have mentioned that there are approximately 1400 genes per cell. Okay. Now the plot 3 is about the mitochondrial distribution. So percentage, the x-axis is the percentage of mitochondrial genes and the number of cells. So good range is about less than 20% for the healthy cells, and median is around 6%, which is absolutely fine with your data set, which is a very good. This is an excellent data set. Okay. So why these plots matter? These 3 plots are your quality control dashboard. They tell you immediately if your experiment worked and guide filtering decisions. If you put bad data in, then the results will be bad. So if you have a good quality control metrics or good data, then your analysis will be perfectly fine. Okay, these are the key metrics that you need to focus on. Next, we go for the filtering strategy. So we are using a quantile approach. So instead of for arbitrary filtering thresholds, we are using data-driven percentiles over here. So these are the percentiles, and this approach adapts to each data set automatically. So you don't have to worry about like remove cells with less than 500 genes or something like that. So we are using the quantile approach. So this is a very good strategy to filter out the cells. So cells before filtering were 55,000, but cells after filtering were 49,000. And genes which were 33,000, after filtering they came down to 18,000 or 19,000 approximately. So it removed 5,545 cells and 14,678 genes, which are unnecessary for your data sets, because we are focusing on CD8 plus data set. Okay, this filtering strategy is also an important and key step to identify high quality metrics. We removed empty droplets and doublets, and genes expressed in less than 0.01% of the cells aren't informative, because those are not informative in our scenario. And quality over quantity, better to analyze 47K high quality cells than 55K mixed quality cells, which has doublets and which has empty droplets. So we have removed all of them together. Okay, and polars makes it much more faster. Next, we go for the pre-processing pipeline. So normally what we do is we normalize the data and find the most informative genes, the ones that vary between the cell types. Some cells captured more RNA than others. The solution is to scale all the cells to 10,000 total counts, then log transform them. So this is the normalization procedure, where you normalize the cells to 10,000 counts and then log normalize it. That's how you identify the highly variable genes detections. This plot that you get over here, on the x-axis you have the mean expression, which is log transformed, and this is your normalized dispersion on the y-axis. The gray dots are the regular genes, and the red dots are your highly variable genes. So these highly variable genes are the stars of the single cell analysis. These are the genes that are turned on in some cells but off in others. Exactly what we need to identify different cell types. The gray ones are the housekeeping genes, which are similar in all the cells, which expressed similarly in all the cells. So we have around 3000 variable genes, as you can see from here. After analyzing 49,661 cells, we have 3000 variable genes from a total of 28,000 genes that we identified after filtering. So this reduces the noise while keeping all the biologically relevant information. It's like filtering signal from the noise. Okay. Next, we move on to the dimensionality reduction. We need some mathematical magic here. So we are using PCA, principal component analysis. So what PCA does is it takes input of these 3000 highly variable genes, the gene dimensions per cell, and output 50 principal components. Okay. The goal here is to capture maximum variation in minimum dimensions. So what we are looking here is a two-dimensional plot, and you can see that we are inputting 49,000 cells. Each dot represents a cell. So the colors here that represents the total counts. Darker means more RNA captured, and the spread that you are looking here shows cell-to-cell variations. And clusters, and these are some of the clusters that you see here, 1 and 2 clusters that you see over here. They are the early hints of the cell population. And then we are doing UMAP calculations after this. So we found 2 clusters. So why UMAP is important is because it's like a GPS for the cells. So it places similar cells close together and different cells far apart. So it's the gold standard for single cell visualization. As you can see, we have 2 UMAPs, 2 colored cell populations. Okay, that's why UMAP is very, very important. So we both did PCA and the UMAP for our filtered data set to identify initial clusters from the cell population. Next, we go for the cell clustering. So we'll use the Leiden algorithm over here to automatically group all the similar cells into cluster. Each cluster potentially represents a different cell type or a state. Okay, so as you can see over here, you have the cluster statistics. So in total, we have 22 clusters identified, 23 in fact, 23 clusters identified, because it starts with 0 to 22, 23 clusters. N cells represents the number of cells, okay, the size of each population, each cluster. And the mean counts represents your RNA abundance or the activity level. The mean genes represents the transcriptional complexity, and your mean mitochondrial percentage is your stress or health indicator. Whether they are dying cells or they are healthy cells, it will indicate that particular statistics. Okay. Notice how different clusters have different characteristics. Some clusters have higher gene counts, more active. Others have different mitochondrial percentage, different metabolic states. This suggests that we are capturing real biological differences, by using this particular clustering algorithm. So in total, we have identified 23 clusters. And then we go for the UMAP visualizations. So all these clusters that we identified, we need to put them in a UMAP coordinates so that you can see exactly how these clusters have been identified. Remember this cluster 0 to 22, as you can see, all different types of colorings, and your UMAP is ready to be visualized. So each dot represents the CD8 plus T-cell diversity. Each colored region represents a different subtype, possibly a naive T-cells, memory T-cells, effector T-cells, or exhausted T-cells. So this beautiful separation shows our analysis is working perfectly. And then next we move on to the marker analysis, where you see all the CD8 plus T-cell markers. So generally the CD8 marker genes that we know already are these genes. These marker genes are well known, well studied, so you can take out all those T-cell markers to check what type of cell population that you have. from those clusters. So here is the plot of those CD8 plus markers, where you can see the expression levels, the mean expressions level. The red, the darker ones are the highly expressed, and the lighter ones are low expressed within these 23 clusters. Okay, so these are the CD8 plus T-cell markers expressions by the cluster. And why I'm particularly focusing on ZZMB is because it kills the tumors of a cancer cell. And CCR7 plus, which is the gold standard marker of CD8 plus T cells, is that it provides a long-term protection. We are looking at the cellular army that protects us from the disease. So these are the main cell markers. Okay, so the final summary is that we have the original data set of 55,000 and 33,000 genes. After QC filtering, we came out to 49,000 to 18,000. Highly variable genes, out of 49,000 cells, we have 3,000 genes. Final clusters were 23, and the cells analyzed were around 49,000 cells. And the key results that we identified 23 distinct cell population, and the average UMI counts per cell is 4,000, the median value. Average genes per cell is around 1400. Mitochondrial gene percentage is around 8.6%. And there are CD8 plus T cell markers detected with 6, because we provided all the published CD8 plus T cell markers. And we have used these beautiful modern stack Python packages to make it ready for your analysis. And this particular notebook will be available on my Omixium YouTube channel. You go to the single cell analysis and you find the analysis.py file with the requirements, and I have prepared a guide also. This was an old guide, but you can also read about this and can plot different types of plots that you require. If you want to analyze in a deeper sense, if you want to analyze even more, then you can find out all the codes and analysis that you can run via Scanpy package. And remember to use the latest Python packages so that you can run it faster and even in a very nice manner. So yeah, that's it, and thank you very much. If you like the video, please subscribe to my channel, and please enjoy watching it. Peace.